Have you ever wondered what happens to antibiotics after we use them, not just in hospitals, but on the farm? Let me set the scene. Antibiotics like amoxicillin, a broad-spectrum medication you're probably familiar with from your last doctor's visit, don't just vanish after doing their job. Instead, they often end up in agricultural soils, mainly through manure, wastewater, or contaminated irrigation. This is where the term phytotoxicity comes in. It describes the toxic effects chemicals can have on plants. Now, you might be thinking, but aren't antibiotics supposed to help? Yes, in humans and animals. But in crops, the story is more complicated. When antibiotics accumulate in the soil, they may alter plant growth and even contribute to antibiotic resistance, a real environmental puzzle. Why does this matter? Well, crops like lentils and chickpeas are global staples, feeding millions and fixing nitrogen in the soil process that supports sustainable agriculture. If these plants are affected by residual antibiotics, it's not just a crop problem, it's a food security issue. The technical term antibiotic uptake describes how plants absorb these substances from their environment. Imagine the chain, from prescription pads to your dinner plate. The question becomes, how much is too much, and which crops are at risk? Picture a laboratory. Rows of petri dishes lined up with seedsome lentils, some chickpeas. Researchers at Damascus University set out to answer a pressing question. How do different concentrations of amoxicillin affect the germination and early growth of these vital crops? They used a completely randomized design, or CRD. Think of it as a scientific lottery, ensuring each plant gets a fair shot at each treatment. The seeds were exposed to amoxicillin concentrations from 0 mg, L, that's our control, all the way to 1000 mg, L. Now, you might ask, why focus on germination and seedlings? Here's the rationale. Early growth stages are like a plant's childhood-y setback here can echo through its entire life cycle. Two technical terms pop up. Root elongation, how much a root grows, and shoot length, the above-ground stem growth. Both are sensitive indicators of plant health. The setup. 6 lentil and 2 chickpea varieties, 3 replicates each, and systematic measurements of root, shoot, and total length. So, could these tiny seeds weather an amoxicillin storm? Let's look at what the data revealed. Let's break down what happened. The headline? Seed germination rates were largely unaffected even at the highest amoxicillin concentrations. Surprised? I was too. Germination is a conserved process, a technical way of saying it's robust and hard to disrupt. But here comes the twist. When it comes to root and shoot growth, the story changes dramatically. As amoxicillin concentration increased, both root length and shoot length dropped sometimes by over 50%. The technical term percentage reduction was used to compare how much shorter roots and shoots became compared to untreated controls. Now, imagine you're a plant breeder. Some varieties like Idlib 4 lentil and Ghab 3 chickpea stood out for their tolerance, showing the smallest reductions in both root and total seedling length. Meanwhile, others saw roots shrink dramatically. Why is root sensitivity so crucial? Because roots are the plant's front line, absorbing water and nutrients. A stunted root system means a weaker plant, less resilient to drought, and poorer yields. So, the key question for agriculture: can we select or engineer crops that shrug off antibiotic residues? What does all this mean for our food and farming systems? First, antibiotic residues in soil aren't just a theoretical concern. They can slow down plant growth, reduce crop yields, and potentially introduce antibiotics into the food chain, a process called bioaccumulation. That's when substances build up in living organisms over time. Now, you might be thinking, if germination is safe, is everything else okay? Not quite. The early seedling stage sets the stage for the entire crop, and if roots or shoots are compromised, the whole plant suffers. Here's where antibiotic resistance, the ability of microbes to survive exposure, enters the picture. When plants absorb antibiotics, they can create microenvironments in the soil that foster resistant bacteria, which could ultimately impact human health. So, what's the solution? Better wastewater treatment, careful use of manure, and breeding or selecting crop varieties with higher tolerance like Idlib 4 and Ghab 3 could help. Ultimately, understanding and managing antibiotic impacts means safer food, healthier soils, and a step toward truly sustainable agriculture. So next time you think about antibiotics, remember, their story doesn't end at the pharmacy.